Hello student, good evening. Myself Sri K.D. Borse sir, biology subject teacher. In last lecture we detail discuss molecular basis of inheritance chapter content concepts, regulation of gene expression, concept of operon and lac operon. Today we'll learn some more concepts include molecular basis of inheritance chapter, genomics, concept of genome and human genome project. So let us start today's first concept, genomics. In genomics, first term include what is genome? The total genetic constitution of an organism called as genome. What is genome? The total genetic constitution of an organism called as genome. The term genome was introduced by H. Winkler, who introduced the term genome. H. Winkler in 1920. Always remember, genome term introduced by H. Winkler. And what is genome? Total genetic constitution of an organism. Alternatively, it is a complete copy of genetic information, DNA, or a complete set of chromosomes are also called as genome. Monad or haploid of an organism. What is genomic? Study of genome through analysis, sequencing and mapping of the gene. Study of genome through analysis, sequencing and mapping of the gene along with their functions, such a branch called as genomics. Means in genomic branch, study of genome with analysis, sequencing and mapping of genes with their function study called as genomics. The term genomics coined by T.H. Roderick in 1986. Types of genomics. There are two types of genomics, structural genomics and functional genomics. There are two types of genomics, structural genomics and functional genomics. What is structural genomics? In structural genomics involve mapping, sequencing and analyzing of the genome. Such type of genomic called as structural genomics. Structural genomic contain study mapping of the gene, sequencing of the gene, analyzing of the gene called as structural genomics. What is functional genomics? It deals with study of function of all gene sequences and their expression in an organism. Up to this we detail discuss two types of genomics, structural genomics and functional genomics. Application of genomics. What are the applications of genomics? Genomics used to improve of crop plants. Genomics used to improve or improvement of crop plants, also human health, also livestock. Second application of genomics, the knowledge and understanding acquired by genomics, research can be applied in medicine, biotechnology and social science. Medicine, biotechnology and social science. Genomics help in treatment of genetic disorders. You know genetic disorders. So treatment of genetic disorder is also help genomics through gene therapy. So this is also one of the application of genomic. Now the fourth one is to develop transgenic crops having more desired characters. With the help of genomics, develop transgenic crop having more desired characters. Genetic markers developed in genomics have application in forensic analysis. Genetic marker developed by genomics. Genomic can be leads to introduce new gene in microbe. In the microbe, new gene introduced to produce enzymes, therapeutic proteins and biofuels. Genomics can lead to introduce new gene in the microbes to produce different enzymes, different therapeutic products, proteins and biofuels. Up to this we detail discuss different applications of genomics. Hope that you understood everything. Now we detail discuss Human Genome Project. Human Genome Project was initiated in 1990 under the international administration of the Human Genome Organization. HUGO means Human Genome Organization. This project was coordinated by the US Department of Energy and National Institute of Health. Human Genome Project coordinated by US Department of Energy and National Institute of Health. International partners in the United Kingdoms, France, Germany, Japan, India and China. Means Human Genome Project involve all these countries like France, Germany, Japan, India and China. HGP means Human Genome Project formally started in 1990 and completed was in 2003. A multinational research project. It is a multinational research project because it involves number of countries to determine the genome or genomic structure of human. What is the main aim of Human Genome Project? To determine the genomic structure of human. We learn what are the main aims of the HGP means Human Genome Project. Number one, mapping the entire human genome. Mapping the entire human genome at the level of nucleotide sequences. You know different nucleotides, adenine, guanine, thymine and cytosines are present in human DNA. So entire human genome at the level of nucleotide sequences mapping is one of the main aim of Human Genome Project. Number second, to store the information collected from the project in database. Stored the information collected from the project in database from different countries to develop tools and techniques for analyzing the data. To develop tools and techniques is also one of the main aim of HGP for analyzing the data. Number 4, transfer of related technology. Transfer of related technologies to the private sectors such as industries, taking care of legal, ethical and social issue which may arises from project. This is also one of the most important aim of HGP, taking care of the legal, ethical and social issues which may arises from project. Now, to provide the complete and accurate sequences of the 3 billion DNA base pairs, 3 billion DNA base pairs that make up the human genome. And last, to find out estimated number of human genes. To find out estimated number of human genes, up to this total noted 7 different aims that are the main aims of Human Genome Project. Most of time this question is asked, what are the main aims of Human Genome Project? Now, HGP was closely associated with rapid development of a new area in biology called as bioinformatics. Human Genome Project was closely associated with rapid development in new area in the biology called as bioinformatics. Always remember, bioinformatics is one of the new branch of the biology. What are the significance of Human Genome Project? Number 1, increase knowledge about the function of genes and proteins. Increase knowledge about the function of genes and proteins. Second significance, a major impact in the field like medicine, biotechnology and life science. A major impact of this field, medicine, biotechnology and life science. Increase understanding of gene structure and function of the other species. Increased understanding of gene structure and function of other species. Such study will enhance understanding of human evolution. Up to this, we detailed discuss different significance of Human Genome Project. Comparative genome size of the human and other models of the organism. In the table noted, organisms, chromosome numbers, estimated gene numbers, estimated size base pairs. Number 1, first, human Homo sapiens, 46 number of diploid chromosomes, estimated gene numbers 33,000 and estimated size base pairs 3 billions. Now second one, mouse, 40 diploid chromosomes, estimated number of genes 25,000, estimated size base pair 2.9 billion. Now third, fruit fly, Drosophila melanogaster, 8 diploid chromosomes. Estimated gene numbers 13,000. Estimated base pairs 165 million. Now plant 10 diploid chromosomes, 25,000 estimated gene numbers and estimated size base pairs 157 millions. Roundworm 12 diploid chromosomes, estimated gene numbers 19,000, estimated base pairs 97 million. East Sarcomyces cerevisiae 32 diploid number of chromosomes, estimated gene number 6,000, estimated base pair numbers 12 millions. Last bacteria Escherichia coli diploid chromosome number 1, 4,400 estimated genes, 4.6 million estimated base pairs. So here noted organisms, their diploid number of chromosomes, estimated gene numbers and estimated base pairs. Always remember all these different organisms and their diploid number of chromosomes, their estimated gene numbers and their estimated base pairs. Up to this today's lecture completed. Review today's topic. We learned today genomics is one of the new concept of molecular basis of inheritance chapter. Concept of genome, type of genomics. You know there are two types of genomics, structural genomics and functional genomics. We learn different applications of genomics. We detail discuss today's lecture Human Genome Project. In that, what are the main aims of Human Genome Project, significance of Human Genome Project, comparative genome size of human and other models of organism. On the basis of today's lecture, some exercise question noted here. MCQs questions. Number 1, first, study of mapping, sequencing and analyzing of the genome. Such type of genomics called as functional genomics, structural genomics, cell genomics, both A and B. Yes, the correct answer is structural genomics, B, structural genomics. In that, study of mapping, sequencing and analyzing of genome. Second MCQ, Human Genome Project was closely associated with rapid development of new area in the biology called as genomics, medicinal microbiology, embryology, bioinformatics. Yes, the correct answer is bioinformatics is new area in the biology. Now, third MCQ, structural genomics involve dash of the genome. A, mapping, B, sequencing, C, analyzing, D, all of this. Yes, the correct answer is all of this. In structural genomics involve mapping also, sequencing also, analyzing. Now, short answer questions. What is genome? Total genetic constitution of an organism called as genome. What is genomics? Yes, study of genome through mapping, sequencing and analyzing along with their function, such branch called as genomics. Who coined the term genome? Yes, H. Winkler, scientist name, coined the genome term. What are the types of genomics? There are two types of genomics, structural genomics and functional genomics. The Human Genome Project was initiated in, yes, 1990. What is HUGO? Human Genome Organization, called as HUGO. How many estimated number of genes in human being? Yes, 33,000 estimated genes in human being. Match column A with column B. In column A, mouse, fruit fly, roundworm. In column B, noted diploid number of chromosome 8, diploid number of chromosome 12, diploid number of chromosome 14. First, mouse. Yes, mouse contain diploid number of chromosome 40, means 1 match with C. Fruit fly. Yes, in fruit fly diploid number of chromosomes are 8, so 2 match with A. Roundworm. Yes, roundworm contain diploid number of chromosomes 12, means 3 match with B, 1 match with C, 2 match with A and 3 match with B. Up to this, today's lecture completed. Hope that you understood everything. If you like the video, then you can subscribe my YouTube bioinfo channel, tap the bell icon and like the video. If any doubt about today's lecture, mention in comment box. Thank you.